So let's pick up with cerebrospinal fluid that we left yesterday's class with. Before we get into our discussion of the individual structures, the sections, regions of the brain, we're talking still about the protection and gross organization. We left off with the cerebrospinal fluid, which we identified as a method of chemical and physical protection for the brain. It surrounds the brain and spinal cord. The central nervous system floats within this cavity, floats in the cerebrospinal fluid. And it's always being produced. There's a tight pH balance that's maintained. It's always being produced, and it's always drained to the vasculature. Now, it's always produced by what we call the choroid plexuses, and we'll see, hopefully, in a moment where those lie within the ventricles. Produced within the choroid plexuses in the lateral ventricles, the third ventricle, and the fourth ventricles, and we talked about how the blood is filtered in order to allow the CSF to be produced, and how some hormones, some metabolites can go backwards and be sent to the bloodstream for delivery to the rest of the body, allowing communication between the brain and other organs in a non-neural, non-electrical kind of way. We'll put it that way. So the choroid plexuses are really, they span almost the length of the ventricles, and we can think about the ventricles as a system of conduits. They are all connected, and I've highlighted the obvious sections here where you can see the choroid plexuses. They're little buds coming off of the bottom of the ventricle, or the floor of the ventricle. But these do extend all the way through the floor of the lateral ventricle here, so across the entire floor. We just can't see them right now, in the third and in the fourth ventricle as we get there. So what we want to do is trace the flow of cerebrospinal fluid through the ventricles. So you can follow along with the flow chart on the right, and I'll try to do my best to describe the 3D orientation. The model isn't really working right now, which is unfortunate, but maybe we'll get there. So starting from point number one, the lateral ventricles. There's one in each of the hemispheres of the brain. They are the large C-shaped ventricles that we saw on the last slide. Choroid plexuses here produce cerebrospinal fluid. When they fill the ventricles, it leaks out through the interventricular foramen. One foramina, two foramen, interventricular foramen to the third ventricle, which is central in the middle of the two hemispheres and encompassed by the hypothalamus that we'll see later today. When the third ventricle is filled, passes through the cerebral aqueduct, or the middle aqueduct of the brain, to the fourth ventricle. And from the fourth ventricle, which is not really a ventricle, it's one of the smallest spaces for cerebrospinal fluid in the brain, there are three exit points. There are two lateral exit points and one medial exit point. We call these apertures. Two lateral apertures and one median aperture. It's not immediately obvious, but the median aperture is here at the base, sandwiched between the cerebellum, which is this structure on the left, and then the brainstem spinal cord here. You can see the purple arrows coming out. That's cerebrospinal fluid to circulate around the brain. And what was that space called where the cerebrospinal fluid circulates after it leaves the ventricles? Sorry? No, it passes through the foramina into the space around the brain. And think about the meninges, the layers. It might even be on the slide here. Yeah, it is. Remember the meninges? Outermost, dura, second, arachnoid, third, pia. Does that ring any bells? Subarachnoid space, thank you. The cerebrospinal fluid fills the subarachnoid space to buoy, to float the central nervous system. And it is right on the slide. Subarachnoid space, right over here. The subarachnoid space connects to the venous drainage through the arachnoid villi that we pointed out last class. The cauliflower or broccoli-looking protrusions that you can see here at the top surface here. There's 1, 2 along the top, a third one sort of anterior in the brain, protruding through the dura mater into the the venous drainage of the brain. And this allows filtering or passage of the cerebrospinal fluid back to the venous side to be returned to the heart. It allows for there to be no buildup of pressure in the ventricles. If we're always producing CSF, we need to be able to balance that process out, and we do that through the arachnoid villi. The arachnoid villi filter it to the venous blood. Arterial blood then, after it's reoxygenated, pass all the way back through the body, eventually to the choroid plexuses, and the whole process starts over again. So just highlight a few points on this slide where we can see the choroid plexuses. We can see the first in the lateral ventricles here, the third ventricle in the middle, adds to the cerebrospinal fluid, and a very small plexus in the fourth ventricle, which just looks like an enlarged tube anterior to the cerebellum, part of the brainstem. So that, I think, is, and then the arachnoid villus. That, I think, is an important process to remember as far as the support and protection of the brain. This slide simply gives you another slide to look at, a different orientation to imagine the flow of cerebrospinal fluid through the brain. You can see the lateral ventricles here, the middle cerebral aqueduct, the fourth ventricle. What we're not seeing is the large shape, the substance of the lateral ventricles, or the third ventricle. But another way to imagine the flow of cerebrospinal fluid through the brain. Questions? So far, so good? Okay. Let us turn our attention to the brainstem. I think admittedly, we're not going to be able to cover as much as I would like in this class. We will get through the brainstem and hopefully discuss the cerebellum, but leave the cerebrum till next week. In and of itself, it's a fairly large topic, which means we push back our discussion of the eye, which for those of you in lab on Monday may not be ideal, but all of the information that you need to dissect your eye is in your lab manual. Take that up as review in class next week. But the brainstem, let's start there. Brainstem is the interface between the spinal cord and the brain, the connection point between those two aspects of the central nervous system. The brainstem is also the major source of motor signals and the major relay point for the afferent sensory signals for the head and neck. 10 of our 12 cranial nerves originate from the brainstem. We talked about the cranial nerves being the major nerves controlling the head and neck, movements, articulations, positions, senses, etc. 10 of the 12 originate from the brainstem. The brainstem might be this area, highlighted here in a green circle. And if you imagine, we're looking up from the bottom, sort of on an oblique anterior face of the brainstem. The anterior aspect here is what is responsible for taking motor information out to the tissues. and the posterior aspect brings sensory information back to be processed or to be a reflex and sent back out again. So posterior, afferent signals, senses coming from the periphery. Anterior is motor. And there are three substructures within the brainstem you can see listed here: the medulla oblongata, the pons, and the midbrain, which we'll discuss each in turn briefly. So starting from the base and working our way up, medulla oblongata is first, the lowest portion with immediate connection to the spinal cord. And here we're looking at it again from an anterior point of view, but also a supero-anterior point of view through the transverse slicing of the medulla oblongata. So it's continuous with the spinal cord, contains both motor and sensory tracts, like we talked about. Motor generally in the front, sensory in the back. And this is largely responsible, or has a large role, in regulating autonomic nervous function, respiration, heart rate, blood pressure. There are other centers that we'll talk about today that also play a large role in regulating those autonomic functions, but this has the first major role in autonomic nervous function. We have 4 of the cranial nerves originating here. You see a few of them on this slide. I'm not even going to mention them because that's our focus of next week. And you can see a few of the names that have Roman numerals beside them that should become familiar as we talk about those in depth. And the structure of the medulla is, at first glance, to have 4 ridges on the anterior face. So this image partly reveals the underlying structures. You can imagine those ridges if you look to the right-hand side. So we have one ridge here with this bulge on the right-hand side. A second ridge is more of a column down the middle on the right border. And those would be repeated on the left-hand side if they hadn't been removed, just for ease of picturing the medulla oblongata. Those ridges, there are 2 pairs of ridges. The first 2 down the middle are the pyramids. You can see that labeled accordingly. And the olives, the medullary olives, are the bulges on the right-hand side. Those must have been named a bit more recently when we got away from the Latin naming and overly complicated descriptors in the names. They look like olives. Let's call them olives. The pyramids are responsible for sending the motor information from the cerebrum. These are the tracts for motor signals coming from the cerebrum. Like railroad tracks, the tracts with T. These send motor input out. You can see there's a brief crossing here, which partly explains why regions of the brain on one hemisphere control the contralateral muscles. It's this crossing over, the decussation of pyramids. And the olives extend projections, you can't see it here, but into the cerebellum, which we'll talk about next, that help you modify the motor action to learn new skills. Modify information to pick up and learn, practice new skills so that they become second nature. They have a large role in the process of instruction or learning new skills to fine-tune the motor outputs to achieve whatever task you're trying to learn. And there will be a complementary slide like this that summarizes most of what we talked about already for each of these regions. Here we're only listing 2 of the cranial nerves. What's missing are the hypoglossal accessory, vestibulocochlear, and glossopharyngeal. Those are the 4 that come off of this section, but you don't need to know that yet. The 2 structural regions The pyramids and the olives are the two structures that we want to pay attention to in this region. And while there's a large role here for autonomic nervous control, again, respiration, heart rate, other characteristics can be traced back here too. The vomiting reflex, hiccuping reflex seem to perhaps not originate, but largely be dictated by actions of the medulla oblongata. Let's leave our discussion there for now. Any questions about this structure? So far, so good. I admit, a lot of what we're going to talk about today is going to be receives sensory information from the periphery, helps to coordinate breathing and heart rate and blood pressure. A lot of these structures do control or are involved in those processes. Yeah, question?
Was it the posterior or the anterior side of the medulla that controls motor versus respiration?
Good question. Posterior or the anterior side that controls motor action. It's the anterior side, and if you humor me, I'll flip back quickly. We can see the anterior side here with the pyramids that are the motor tracts. Good question. The anterior side sends motor signals. Okay. Let's move up. So from the spinal cord to the medulla oblongata, next we reach the pons, which literally translates as bridge. This is a major relay station for signals returning from the periphery and being sent out from the cerebrum to the rest of the body, a bridge between these two regions. Now it acts as a bridge between the brain and the body, but also between hemispheres of the brain, so left to right. And because of where it's situated, you can see the vertical position here shown nicely on the sagittal slide. Because of where it sits vertically, it's also a relay point between the cerebellum, just posterior to the pons, and the cerebrum above it. The pons is this obvious prominence on the brain stem. You look at a brain stem, you know nothing about it, you see a big bulge in the brain stem, that's the pons. It's really easy to identify. 4 more cerebral nerves or cranial nerves originate here, and we're over halfway, having only gotten through 2 substructures. Large majority of motor output and sensory innervation in these even small 2 substructures of the brainstem. I won't talk at length about its role. I will say that this does also control respiration, but few other processes. The ventral portion does assist the medulla in controlling respiration or monitoring the inhale, the depth of the inhale, the pacing of the inhale, and then helping to transition to an exhale at the lungs. I'll talk more about that on this slide. So 4 other nerves that arise here. This is a primer for our discussion next week. These names, these words that make no sense now will become obvious and commonplace. They'll be household names in your notebooks, in your reviews, in these videos. You'll know them well. There are really fun ways that we often can tell you to help you remember them well, but I digress. That's down the road. So major relay station, and the control of respiration occurs in this lower point, the pneumotaxic and the apneustic areas of the pons help control respiration. And so specifically, the pneumotaxic area prevents overinflation of the lungs. It detects when the respiratory muscles are stretched to near their limits, or the lungs are stretched to the limits of the thoracic cavity, and it prevents your body from trying to take a breath that's too deep, that wouldn't be allowed by the physical constraints of the thorax. So it prevents overinflation of the lungs and damage to the lungs. And then in combination with the apneustic area, controls the transition between breaths. We don't ever really think about it. You just take a breath in and it goes out, in, out. But you need to activate and then deactivate muscles in quick succession. And then the contralateral muscles need to have the opposite activation and deactivation to ensure successful movement of air from the environment into the lungs. So it smooths the transition between breaths. And not just smooths the transition, but makes sure that we don't pause too long. Helps with timing of the breath. And then in a complementary fashion to the pneumotaxic area, helps to make sure that we don't breathe out too much. So these two areas are important in controlling the level of respiration, the points to which we breathe in or breathe out. And I'd say on average they do a pretty good job, right? They must. We take it for granted, but on average you take 5,000 breaths a day perhaps. On average, the inhales equal the exhales. You stay the same size as you were when you woke up. You don't inflate or deflate. On average, they're pretty good at controlling respiration. So that's what we want to know about the Pons. Relay station, some control in helping to execute smooth breathing. Oh, what time is it? Nothing important. Let's move on up. From the medulla, and often the medulla oblongata is called the medulla in short, just so we're clear. To the pons, next we find the midbrain, superior to the medulla oblongata, and extends from the pons to the diencephalon, whatever that is. We don't know. We'll find out shortly. If the pons is the relay station, this is the checkpoint for outgoing signals from the cerebrum to the relay station. It can filter out some things that shouldn't be relayed. It also has a really important role in automatic reflexes. Now I'll highlight the area here, the midbrain, and the areas involved in reflexes are specifically on the tectum. Tectum is the surface, and these little round bulbs are colliculi. We call them the superior and the inferior colliculi. Those are specifically involved in registering and reacting to automatic reflexes. Reflexes that are below conscious awareness. Here, below conscious awareness means physically below, as in inferior to. But also the reflexes never arise within your train of thought. So what reflexes are we talking about? The superior colliculi deal specifically with visual signals and then regulate head and neck movement, also maybe body movement if it's a large visual signal. So flashes in the periphery, or you see a really fast-moving object, a baseball or a basketball coming from the periphery. This is involved in the flinch response to get out of the way. Whereas the inferior colliculi respond to auditory signals. We're not in a major urban center, so it's not likely that we'll respond to gunshots, but large bangs like firecrackers, perhaps. if you're out hunting, a shotgun blast, cymbals crashing, something falling. Those are the types of responses that the colliculi respond to, or are reflexive of. No surprise, we also have some heart rate and blood pressure regulation here. Importantly, all of these that we've talked about, all of these subsections, these levels are below conscious awareness and automatic, which is good because you don't then need to regulate heart rate and blood pressure and respiration while you're asleep and unconscious. Really important that we make that division. The midbrain is also a really important region for connection with the cerebellum. And the cerebellum is largely tasked with planning and executing movement of skeletal muscle. And so relays and signals to the cerebellum from the midbrain are involved in smooth movements, planning smooth movements. And those two areas specifically that I'm thinking of are the substantia nigra and the red nuclei. Now it's worth mentioning too, the first example in this list, the corpora quadrigemini, is just another word for the collection of superior and inferior colliculi. There's 4 of them. So the old name was the corpora quadrigemini. An important feature of the midbrain. But here, deep, not on the surface of the midbrain, deep within the midbrain, we can see these other structures. The substantia nigra, this darkened area. There are 2 aspects on both sides of the midbrain. And then the red nuclei, fairly obvious, red nuclei here that are so named for the iron-rich content of the cell bodies in that region. So the substantia nigra specifically have a large proportion of neurons that release or respond to dopamine. And dopamine is a reward neurotransmitter. In learning or in competing, dopamine is often released to reward you with having achieved a positive outcome. We like dopamine. It's a mechanism that a lot of addictive substances work as well. This region is involved in coordination of smooth movements, and it's loss of neurons here that is related to the onset of Parkinson's disease, which is characterized by a loss of smooth, controlled movement of the muscles. Now, a lot of these nervous signals will pass through and be assisted by the red nuclei as well, so they have that combined function to help process and plan smooth movements. And it's hard to imagine what that means at first, because you're in your kitchen and you open the cupboards and you reach for a can of soup and your arm just goes up. And you can target that visually and you know how far to reach, and you can grab onto it and you can pick it up and you bring it out, move on about cooking your soup. But the planning that's required to execute that directly, I think it's underappreciated. We often overlook it. So someone that might lose that coordinated planning will see the soup can and want to grab it, but their plan of action won't be linear. It won't be smooth and coordinated, and then the grasping is often off the mark. That type of streamlining occurs here. So major structures that at least I think it's important to look at in the midbrain. Again, not an exhaustive list. Any more questions so far? I see a lot of really excited faces, writing notes down, some that are distracted, but that's okay. You check in and out. I get it. It's hard to sit here for 50 minutes. Anything you missed, though, in the distractions? No? You're waiting to get the YouTube video. Okay. All right, let's move up. Maybe not up. Maybe in. This is an addendum. This is part of the brainstem, but it wasn't a formal subsection as listed in the initial list. The reticular formation, or specifically the reticular ascending system, also known as the reticulating or reticular activating system. We're going to call it RAS because it's easy to remember. And I don't know why this keeps cutting out. I apologize. Luckily, it keeps coming back too. If that stops, we're in trouble. So the RAS, the reticular ascending activating system, is really responsible for maintaining arousal. And not arousal like you're thinking of, but consciousness. Are you awake and paying attention right now? Activating the RAS is what is responsible for you being awake and paying attention. So it receives sensory information from the eyes, the ears, many special senses, that is delivered to the higher order centers in the brain to maintain and support awareness and consciousness of those senses. I'd like to say the majority of us have full RAS activation right now. Some of you do. Some of you are in the middle somewhere. Inactivation occurs when you sleep, which is really good because you don't want to be interrupted by small sounds in the night or smells perhaps. Large ones, yes, can filter through and cause you to wake up, but we inactivate this system to prevent those sensory signals from reaching our awareness so that we can sleep. I appreciate that you're trying to keep this activated right now, which is good. This is a site where melatonin works. You've heard of melatonin. It helps you regulate your circadian cycle, your circadian rhythm. It's produced in parts of the brain that we'll talk about later. It activates, or rather deactivates, the RAS to help you fall asleep. Luckily, it's only deactivated when we sleep. If it's ever damaged, that's characteristic of entering a coma. Light damage to the RAS means light coma that you might wake up from. Any significant damage can cause what we would think of as a deep coma, where you even lose reflexes to maintain heart rate and blood pressure, and you need to put on some system, some external system to keep you alive. Significant damage to this structure is responsible for that. So filtering sensory information and delivering it to the higher order thinking centers. And that's all I want to talk about in the brainstem. I know, you're thinking, well, there's so much more on the slides. I want to know more. Can we go into more detail? No, we'll leave it there, okay? We're going to move instead to the cerebellum. This is more detail, but not about the brainstem. Cerebellum. I referred to this a few times. It kind of looks like a small brain. It's obvious where it is when you see a section or a surface image of the central nervous system. It's posterior and inferior within the cranial cavity. And a lot like the brain, it has two hemispheres, left and right, two lateral hemispheres, and a central, what is called vermis, which simply means worm, because it looks like a worm. central vermis. You'll notice it looks a lot different than some structures in the brainstem, for instance. It looks a lot busier, right? And it is. The cerebellum might be a tenth of brain volume, but probably contains half the neurons in the brain. It's packed densely with neurons. And to be packed densely with neurons, it takes on this special conformation, which is mirrored by the larger cerebrum. The special conformation is this series of really wavy ridges and folds which serve to just increase the surface area of the cerebellum. What's the point of increasing the surface area of the cerebellum? Well, along the surface is where all the neuronal cell bodies live. Now, we'll talk about the difference between gray matter and white matter, I think next week. But in very simple terms, gray matter are cell bodies, white matter are the axons, the tracts, where the signals are sent. So all the processing, the firing, the ATP consuming processes occur in the cell bodies or in the gray matter. Largely representative of a lot of processing or actions going on. And then the white matter are tracks or carrying of signals somewhere else where they need to be acted upon. So the cerebellum has a large surface area full of neuronal cell bodies so it can make a lot of calculations or do a lot of processing. A lot of integrating happens in this organ or this part of the brain. Is it cold season? I've known a few coughs going on. Let's take a break. Let's all just cough. How about that? All right, everyone clear your throat. Take a break. There you go. Let's do some coughing. All right. Get it all out. You don't want to be the person that's just coughing by themselves. We'll all do it. All right. That was a fantastic cough, by the way. I loved it. All right. Rasses are activated. Good. Okay, you guys are awake and conscious. All right, let's keep going. Actually, I had a professor once that in classes like this, not 50-minute classes, but we had hour-and-a-half classes in undergrad, in an auditorium like this, would do the wave with us. He'd go like front to back or side to side or middle out. And I thought about doing that with you as a group, but there's not, the density is a bit lacking on the sides, and everyone has laptops, which is not really wave friendly. So, coughing's good enough. Let's do coughing. I'll have to learn how to edit these videos now. Leave it in, sure. Yeah, why not? Okay. Cerebellum. So we talked about the folds, the large surface area. What's its role? Why does it have such a large surface area? Why is it seemingly so important? Cerebellum is largely tasked with conscious and unconscious movement of skeletal muscle. Movement of muscles, maybe, to not be so specific. So the execution of movement is a very important characteristic that we have as an organism, and this is devoted mostly to that. Now I want to draw your attention to the white area here on the right. This is the inferior view, and it's kind of looking at it from the front a little bit as well, from the anterior side. After spending all that time talking about gray matter, I want to draw your attention to the white matter tracts, which we call peduncles, which is a fun name. So the peduncles from the cerebellum are where it attaches to the brainstem. And they are the roots of what we call the arbor vitae, and this will be clear on the next slide. If you were to slice one of the lobes or halves of the cerebellum in half, you'd see all the gray matter around the outside that culminate, that meet in what look like roots, white roots at the center of the cerebellum. And it looks like a tree. So we call it arbor vitae. These are the endpoints of the arbor vitae, the roots. So this is where all the signals from the massive processing power of the cerebellum are leaving. This is where they're being sent out of the peduncles. So three general regions, the superior, middle, and inferior. The superior section sends signals to the red nuclei. So this is where we have that smooth movement of muscle plan being delivered. The middle is probably the largest portion of the root. And this is where signals associated with voluntary contraction, voluntary movement originate or are sent out. So the somatic system that allows you to think, I'm going to lift my arm, and then do that. Those signals come out here. And then the inferior, largely sensory, so afferent signals enter here. Afferent signals connected to the vestibular system, so position of the head in space. These are signals that largely come from the inner ear, and we'll get to that when we talk about special senses, but it helps to maintain equilibrium. You might know of it as equilibrium or balance. Also, afferent sensory signals from the limbs, muscles of the limbs. Information on the position of your limbs in space arrives here. So if you were to close your eyes and lift your arm up, you wouldn't need to see that your arm was raised to know that it was raised. That information is being sent back from the limb already. We call that proprioception. Also in this region is where we have the olives from the medulla sending information to the cerebellum that help it learn and refine new tasks, help it in task acquisition. So the inferior tract here of the cerebellar peduncle is largely the spot where information comes in, where we receive sensory information. All of that, well, if you put it all together, it makes sense that this is largely to help maintain unconscious control of things like balance. You don't fall over in your seat. I don't have to think about maintaining a posture upright on stage. I learned how to do that when I was 2, maybe 3, I don't know. But if I were slacklining, that would be a different thing. Not automatic. So the cerebellum controls the coordination of skeletal muscle, muscle contractions, and the maintenance of posture. Muscle tone, muscle firing, muscle activation and inactivation to keep us upright. I'll also draw your attention briefly to the arbor vitae, which you can see here nicely, with the cross-section through the cerebellum and all the gray matter around the outside with the many folds and the large surface area. The neuronal cell bodies live there, and the signals from those cell bodies meet and converge in these white matter tracts, which leave via the peduncle, which is this connection here, right posterior to the fourth ventricle, containing the cerebrospinal fluid in the brain. So hopefully these pieces are starting to come together a little bit. You can see that it's very close to the medulla and the pons and the midbrain, and the associated connections are logical. We've highlighted a few of those already today. Let's leave it there.